Good morning, good afternoon, or good evening, wherever you're at, watching this live event number 5, the first one in the epidemiology module of the IAC Summer School. We have our 36 participants attending in the Zoom, and we have many registrants and viewers on YouTube. So welcome to everyone. Our chair today is Dr. Pietro Ferrari, acting head of the Nutrition and Metabolism branch at IAC. And I will invite viewers on YouTube to write their question in the chat, and we will try to address them all after the presentation, of course. Pietro, the floor is yours. Thanks.
Thank you, Sandrine. And hi, everyone. So it's a pleasure today and an honor to introduce Professor Marc Gunter to give a seminar within the activities of the IAC Summer School on cancer epidemiology. So Marc is a friend of the agency. He's currently a professor and chair in cancer epidemiology and prevention at the Department of Epidemiology and Biostatistics, the School of Public Health at Imperial College in London. He obtained a PhD in Cambridge and spent time as a postdoctoral scientist at the National Cancer Institute in the United States. And he is the former branch head of this very same branch, the Nutrition and Metabolism, and he spent time at IAC between 2016 and 2023. So he just left the agency. His research interest span from cancer epidemiology, molecular epidemiology, biochemistry, nutrition, and metabolism. And I would say that despite his relatively young career, Marc authored several hundred papers, some of which were published in prestigious journals. And we're talking about journals with large impact factors. Marc is also leading international consortia, including the European Prospective Investigation into Cancer and Nutrition, and the Discern study, which is a fairly large consortium and an investigation of 3 poorly understood cancer, including kidney, pancreatic, and colorectal cancer. So in this seminar, Professor Gunter will provide a broad overview of the current understanding of the role of diet in cancer, particularly how we study the relationship between diet and nutrition and cancer risk in cancer epidemiology. And then this is a field that is recognized to be very complex, challenging, and sometimes even controversial. So, Marc, thank you for being with us, and the floor is yours.
Okay, thank you, Pietro, for the very nice introduction, and hello to everybody. So I'm just going to share my slides. So can you see my slides? Can you hear me okay?
Yes.
Yes.
Okay. So thank you for the introduction, and hello to all the participants of the IAC Summer School. So I'm going to be talking today about diet and cancer. And as Pietro alluded to, this is a very challenging and complex area, but also very important. We know that obviously diet and nutrition is critical for maintaining health and normal physiology. And there's been this suspected link between nutrition and diet and cancer for many decades now, but actually pinpointing the aspects of the diet and nutrition which are causally associated with cancer has been very challenging. So in the next half an hour or so, I'm going to give a very broad overview of the field, some of the early observations around diet and cancer, what we understand in terms of its impact on the cancer burden. I'm going to present some of the work we've been doing within the EPIC cohort, which Pietro just mentioned, as a resource for studying the link between diet and cancer and other health outcomes. And then at the end, I'm going to move on to talking about some of the more emerging work we're doing within the field. And also I'm going to be talking a bit about obesity and cancer, because obviously that's inextricably linked to nutrition. It's a very important aspect of the work we're doing around diet and cancer. The learning objectives are firstly just to give a very short introduction about the impact of diet on cancer burden. I'll be talking about some of the earlier studies, migrant ecological studies, which gave some clues as to the role of diet in cancer, and then some of the challenges in the field. I'll then talk about some examples from the EPIC study, and why EPIC was set up to study the link between nutrition and cancer. As I said, I'll be briefly mentioning obesity and cancer, and then also emerging hypotheses and advances in diet and cancer. And very happy to take any questions. If things occur to you as I'm talking, please write them in the chat, and then we can address them in the question and answer session at the end. So the hunt for the causes of cancer has been a major topic of debate for many decades now. This was some of the work from Don and Peter back in the early 1980s, where they tried to apportion population attributable risks to different risk factors for cancer and estimate the contributing role for these different risk factors in the cancer burden. And as you can see here, they kind of estimated that maybe around 35% of the total population attributable risks of cancer could be attributed to diet. But notably, they put very wide, almost confidence intervals around this estimate. So they estimated that this could be anything between 10 and 70%. And some of this really reflects differences between cancers. So we know that some cancers are more likely to be related to diet than others. So I think they estimated at that time that maybe around 80 to 90% of stomach cancer could be caused by diet, or around 50% of colorectal cancer. Now we know that those estimates were very much inflated now. They're very inaccurate. And actual proportions probably likely much less, particularly for stomach cancer. But we're still trying to think about what is the contribution of diet to the overall cancer burden. And this has been a source of research for several decades now. Some early clues that diet or even the environment were playing a role in cancer causation came from some migrant studies looking at Japanese migrants moving to Hawaii. This was back in the 1950s, 1960s. And this was work that was led by Larry Colonel and colleagues in Hawaii. And what they did was look at the number of cancer cases occurring among Japanese migrants to Hawaii, and then compared them to the cancer rates for both colon and stomach cancer both in Japan and also more broadly in the US. And what they observed was that Japanese families moving to Hawaii, the rates of colorectal cancer among those migrants increased substantially once they moved to Hawaii, and almost approximated those of the population already in Hawaii at that time. So you can see that for colon cancer, the rates increased substantially. So several decades ago, Japan had one of the lowest rates of colorectal cancer in the world. That's changed a lot now. It now has one of the highest rates. But at that time, had very low rates. So they showed that Japanese migrants moving from Japan, which had very low rates of colon cancer, once they moved to the US, their rates increased substantially. So really suggesting that there was something in the environment or their lifestyle which changed, which then led to the increased risk of colorectal cancer in that population, because it was too early for their genetics to change. Conversely, for stomach cancer, so stomach cancer was very common cancer in Japan at that time, but was relatively or less common in the US. But again, once these Japanese migrants moved to Hawaii, their rates of stomach cancer came down quite considerably. So again, suggesting that something probably shifted in their environment, which then led to that change in incidence rates. So whether that was diet or other lifestyle factors or other environmental exposures wasn't clear, and I still think it isn't entirely clear, but it's highly likely that lifestyle and change in lifestyle and diet quite likely plays a role in this shift. There were some very early ecological studies of diet and cancer that were conducted, which gave some clues as to potential components of the diet which might be driving cancer. So this was an ecologic study looking at rates of colon cancer and meat consumption in different countries of the world. So as you know, I mean, you're on the summer school and you're learning about different types of study design. So these ecological studies, they can't determine causality, but they can give some interesting clues as to what's going on in different populations. So if you look at rates of colon cancer per 100,000 people here on the y-axis, plotted against grams of red meat consumption on the x-axis by country, you can see quite a nice linear trend. For countries which are consuming more meat, they have higher rates of colon cancer. So New Zealand, US, Canada, then from the European countries, in contrast to those consuming much less red meat. So countries in Africa, South America, Japan, at that time, this was several decades ago now, had much lower rates, so suggesting that what this did was to provide some early clues or evidence that maybe red meat consumption could be underlying this variation in colon cancer rates between different countries. Now, moving forward several decades, I mean, very large prospective cohort studies and also before that case control studies have specifically investigated the association between diet and lifestyle and cancer, producing a very large body of evidence. And organizations such as the World Cancer Research Fund and the American Institute of Cancer Research, they have a specific focus on trying to collate these data and put them together and try to come up with more accurate estimates and then recommendations around the role of diet and lifestyle in cancer. This is part of their CUP Global program. Within that program, they've also been trying to estimate population mortality fractions for potential preventability of different cancers. And this is some work they published several years ago now for breast, liver, endometrial, and colorectal cancer. So looking at a proportion of cancers which could have been avoided if people had consumed, for example, a healthier diet or avoided alcohol or maintained a healthier weight or more physically active. So you can see here, if we look on the top left for breast cancer, for 4 countries, US, UK, Brazil, and China. If you look at body fatness, which we know is a causal risk factor for at least postmenopausal breast cancers, you can see that quite a proportion of breast cancer cases could have been prevented had women maintained a healthier weight during their life during the lifespan. Likewise for activity, but also alcohol. You can see here if women had avoided consuming alcohol, a number of a significant proportion of breast cancer cases could have been avoided. What's also notable here is variation between countries, and this really reflects prevalence of that exposure within those countries. So you can see for alcohol, countries such as the UK, which traditionally have consumed more alcohol, the proportion of breast cancer cases attributable to high alcohol consumption is much greater in the UK compared to countries such as Brazil or China, where historically there's been much less consumption in women. But we know some of these patterns of consumption are now changing. So these population mortality factors will likely change in the coming years. We look at endometrial cancer, where we know that obesity is a very strong risk factor for these cancers. In women, you can see that in the US, where obesity has been had a very high prevalence for many years now, more than 50% of endometrial cancer cases could have been avoided if women hadn't become obese. Proportion is also high for UK, and then much less for Brazil and China. But again, because obesity is becoming more prevalent in many countries of the world, including these, this landscape will likely change in the years to come. If you look at some other aspects of diet for colorectal cancer, for example, you can see again body fatness, physical activity also contribute to the burden of colorectal cancer, as well as alcoholic drinks, but also other aspects of the diet. So foods containing fiber, red and processed meat have been linked to a lower risk for fiber. So consuming more fiber lowers risk of colorectal cancer, whereas higher consumption of red and processed meat increases risk of colorectal cancer. So again, proportions vary across countries because of the different prevalence of this consumption of these different aspects of the diet. But again, suggesting that around 15 to 20% of the colorectal cancer burden could be attributed to these aspects of the diet. Now, I just wanted to put this graphic up because this really summarizes current state of the art regarding different aspects of the diet and lifestyle and different cancers which have been evaluated by the World Cancer Research Fund. So each cell, if it's red, it suggests that there's convincing increased risk for this particular aspect of the diet and for that particular cancer. So you can see here for processed meat, there's convincing evidence that it's a likely cause of colorectal cancer. Red meat, also a probable cause of colorectal cancer. And then if you look at some of the green cells, these suggest a convincing evidence of decreased risk. So physical activity, there's convincing evidence that if you engage in more physical activity, you have a low risk of colorectal cancer. We can see for some risk factors, there's quite a lot of evidence suggesting that these risk factors play a role in multiple cancer types. So body fat here, that you can see has been linked to increased risk for a number of different cancers. But what's really striking, I think, about this figure is that there's a lot we don't know. So many of the cells are blank, and that's because there's been a lack of data, or there's inconclusive evidence for the link between these different aspects of the diet and cancer. So this really suggests that we still have quite a lot to learn regarding the impact of diet and lifestyle on cancer. So just to talk about some of the challenges of research on the impact of diet in cancer development. So just to lay out a few points here to keep in mind as we're discussing today. So firstly, nutrition itself is inherently complex. Okay, so we don't consume foods generally on their own. Our diet is a kind of construct of multiple different foods and nutrients, all of which they interact with each other. They interact with the host metabolism. We know that metabolic phenotypes play a role in the degree to which you're exposed to different components of diet. So two people might consume exactly the same diet, but they have different metabolic profiles. They have different gut microbiome profiles, which is another aspect of research which is becoming more and more of interest. The impact of the gut microbiota on cancer, and also whether it's a modifier of the link between diet and cancer. But nutrition itself is inherently complex exposure. It's not like, for example, smoking, where you have a single exposure, which we know has a strong impact on cancer, risk of different cancers. So already there's this background of complexity in the exposure. It's also very difficult and challenging to assess within human studies. So while there's quite a wealth of data linking different aspects of nutrition and tumorigenesis in experimental models, where you're able to control very carefully exposure to particular aspects of the diet or nutrients, doing this in human studies is impossible. And it's actually quite difficult to accurately estimate diet. I mean, this varies depending on the aspect of the diet we're interested in, but exposure assessment error is a major topic within diet and cancer research. How can we get better estimates of habitual diet and overcome some of the measurement limitations which we know are inherent to that? Thirdly, confounding is a major issue. So we know that people who have a particular diet, they also tend to exhibit other behaviors as well, which could be related to cancer. So maybe people who are eating on healthier diets, maybe they're less physically active, maybe they smoke more, maybe they engage in other behaviors which might be related to cancer, and controlling for some of these confounding factors within population-based studies is challenging, because we know that diet is so intricately interwound with other aspects of lifestyle. So being able to control for confounding in nutrition and cancer studies is an important aspect. Fourthly, we don't always understand mechanisms. Okay, so while there's quite consistent evidence, for example, that consuming higher levels of processed meat raises risk of colorectal cancer, the biological mechanisms which underlie that association are not clear. So for many factors of which we've found associations with cancer outcomes, we don't fully understand the underlying biology. And again, this is a weakness, because it's very difficult to explain why we see or observe particular associations if we can't if we don't have good data to sit alongside the epidemiologic evidence from mechanistic studies. It's very difficult to convince people that there is a causal relationship there. And then lastly, we're also generally dealing with very weak to moderate effects. So the degree of the magnitude of association between different aspects of the diet and cancer are not large. We tend to see relative risks in the range of 1.2, 2 if we're lucky. It's not like looking at smoking and lung cancer, where you have very high relative risks. So these weak moderate effects also mean that our studies are very susceptible to the impact of, for example, confounding. We might miss potentially causal associations because of these weak to moderate effects which we see in epidemiologic studies. So this is also a very important point, and this is why we need very large studies and also bringing large studies together, and also triangulation of epidemiologic data with other types of data to infer causality. One area I think many of us are interested in is how can we improve this situation? How can we go beyond just relying on, for example, questionnaire-based data linking nutrition and cancer, and can we make use of other types of research tools? And one area that we're particularly interested in is the application of biomarkers and omics technologies to the nutrition and cancer field to see whether it can help us identify help us to identify more accurate exposures. Can we identify biomarkers of exposure, and also can it help us understand mechanisms linking diet and cancer? So we're particularly interested in prospective cohort studies. A number of these now exist worldwide, and these are really instrumental in studying the link between exposures and cancer outcomes, and particularly for diet, where we can collect information on diet and lifestyle at baseline, so prior to the participants developing cancer. We can collect blood samples, and then we can follow these individuals over many decades, during which time a proportion of them develop cancer. And then we can look back before they developed the cancer and see what was it around their diet or lifestyle or biomarker profiles which meant that they were more likely to develop a particular cancer outcome versus someone who remained healthy. So these are really invaluable tools for research on diet and cancer. And a number of these, as I said, now exist around the world. So I'll be talking a bit about the EPIC study, but we now have UK Biobank, a number of different cohorts in the United States and other regions, Australia, and also in Asia. And these have generally been a really valuable resource for studying diet and its relationship with cancer. As I said, we're particularly interested in also using blood samples and assessing biomarkers of diet and nutrition, and relating those to cancer. And within these cohorts, we collect blood samples generally at baseline, as well as information on diet and lifestyle. And then these blood samples offer a resource for performing various biomarker assays, omics technologies. So, for example, they've been instrumental in looking at the genome-wide association studies, but also now increasingly from metabolomics, proteomics, and also targeted biomarkers, which we can then measure within these blood samples and then link them to cancer outcomes, which have occurred within the cohort. The EPIC study was set up back in the early 90s, a metiac by Elio Riboli and colleagues. And the idea then at that point was to try to capture variation in diet and lifestyle across Europe, and try to relate that to variation in cancer rates, which were observed across different European countries. So this was a very large prospective cohort, recruited more than 520,000 participants across 10 European countries. And follow-up has now been going on for around 20 years. So EPIC has really been a very valuable resource for not only researchers at IARC and the EPIC collaborating centres, but also internationally, and has contributed a really substantial wealth of knowledge to links on diet, and also beyond diet, looking at other risk factors for cancers. So, as I said, the cohort recruited across 10 European countries, really with the aim of trying to capture variation in diet and lifestyle across Western Europe. So recruiting participants in Scandinavia, United Kingdom, Germany, Netherlands, and then France, and then Spain, Italy, and Greece to capture the Mediterranean region. As I said, one of the objectives of EPIC was to try to really capture variation in diet and lifestyle across the cohort. And I think to some extent it was very successful in doing that. This just shows just a snapshot of some of the biomarker work that's been done in EPIC to look at nutritional biomarkers across the European countries within EPIC. So two biomarkers, so lycopene, which is a compound found in vegetables such as tomatoes and red peppers, and then alkyl resorcinol, which is a biomarker of whole grain intake. And what you can see here, if we look at the levels of lycopene in blood across different European or different EPIC centres, you can see the highest levels in the Italian centres and also in Germany and Spain and Southern Europe, and much lower levels in Northern Europe. Conversely, alcohol resorcinols, which are a biomarker of whole grains, tend to be higher in Northern European countries where there's a greater consumption of breads with whole grain and, for example, rye, versus Southern European countries. So this just gives an idea that within EPIC, we were able to capture variation in diet across the different EPIC centres. Also using the questionnaire-based data also allowed us to look at the variation between different EPIC centres and capture variation in diet. So these plots show the degree to which consumption of different aspects of the diet is skewed in different EPIC countries. So this yellow ring marks the average, and then you can see the extent to which different countries have high levels of consumption of different aspects of the diet. So this is data from Greece. So you can see that on average they consume more legumes, vegetables, and vegetable oils compared to the average in EPIC. And if you compare this with Germany, you see a slightly different snapshot. So you can see a greater consumption of coffee, fruits and vegetable juices, more butter as compared to olive oil, and greater levels of processed meat. And then United Kingdom is interesting. You can see that a greater consumption of tea, which was to be expected, but also high levels of soft drinks, and also, again, similar to Germany, greater consumption of milk and butter, and also cake. So, and again, this really reflects the variation in diet across EPIC at baseline. EPIC has been extremely productive. It's produced more than 1,000 scientific publications now and has contributed to important findings regarding diet and cancer, particularly on obesity, fibre, red and processed meat. And where we are in EPIC now is because of the long follow-up and the size of the cohort, we have a large number of cancers which have occurred within the cohort in the last 20 years or so, as well as deaths and other chronic disease endpoints. So this really affords a very exciting opportunity to make use of this data for doing new studies on diet and lifestyle and cancer in the years to come. So to show, for example, different cancers and their incidence or number of cancers within EPIC. So you can see breast cancer is most common, with more than 14,000 incident cases which have occurred since baseline. Colorectal, more than 8,000 cases. But even for less common cases such as kidney, pancreatic, ovarian cancer, we have more than 1,000 incident cases now within the cohort. And what these numbers do is that they allow us not only to look at some of the etiology of these less common cancers, but also the large number of cancers from more common cancers allows us to do more detailed subtype analyses, which we know molecular subtypes and anatomical subtypes are becoming more and more relevant for understanding links between diet and lifestyle and cancer. What's also important to point out, though, is that EPIC has been around now for, as I said, for around 30 years, and diet and lifestyle in Europe has changed during that time. So what we're thinking of trying to do is become less reliant, not just on the baseline data in EPIC, but also try to incorporate data which has been collected during the follow-up period. So just to give an example of why this is important, this is data on or trends in meat and fish consumption in Europe between 1995 and 2011. And what you can see quite clearly is that there's increase in fish and poultry intake during this period, but a reduction in consumption of beef. This was the mid-90s, late 90s was just really when EPIC was getting started. So it's quite likely that many of the participants of EPIC changed their diet and lifestyle during the follow-up period. So it's really important now we try to make use of some of this change in diet and lifestyle within the cohort. And there's a number of initiatives ongoing within EPIC to try to make use of this data which has been collected within the cohort during the follow-up period to look at whether change in lifestyles and diet has had an impact on cancer and other and disease endpoints. We're also trying to move beyond some of the more classical aspects of the diet and think about more innovative ways of classifying diet. One area I think of particular interest is around ultra-processed foods and food processing. So we know that diet in Europe and beyond has become more processed in recent years. And there's been a number of hypotheses which have been advanced to suggest that maybe the consumption of some of the compounds which are present within ultra-processed foods might be playing a role in cancer development. So this hasn't been proven, but it's certainly an area of growing scientific interest. And a cohort with like EPIC, which has very detailed dietary information on a large number of participants, really offers a very exciting platform for looking at some of these newer hypotheses relating diet to cancer. So this is some work that Inge Huberdex has been leading at IARC to try to classify the diet in EPIC depending on the degree of processing of different aspects of different foods, and using the NOVA classification, which groups foods according to, for example, group 1, which is unprocessed or minimally processed foods, versus group 4, which is the ultra-processed foods and drink products. So EPIC has been very busy trying to classify the data in EPIC by the degree of food processing. And we're now at a point where we can look at these different elements of food processing within the diet and relate them to cancer. And as I said, there's a number of different hypotheses linking ultra-processed food intake with cancer. So, for example, we know that ultra-processed foods tend to be obesogenic. They've got lower nutritional value, which itself might have a role in cancer development. But we also know that there are a number of compounds found, so near-formed processing contaminants, some of these dietary advanced glycation end products, and also food additives are all being suggested to potentially play a role in cancer development. To give you a snapshot of some of the results we're getting from EPIC, so this is a paper that was published a couple of months ago now in the Lancet, which looked at food processing and risk of 25 different cancers across EPIC using the NOVA classification. I don't have time to go into the details, but what this generally showed was that greater consumption of the less processed foods, so NOVA group 1, was associated with a lower risk of developing all cancers, but particularly cancers such as head and neck cancer, colorectal cancer, and also liver cancers. Conversely, a greater consumption of more processed foods, so as would be judged by NOVA 3 or NOVA 4, associated with a higher risk of developing these cancers. So what this really suggests is that diets which are richer in more processed food items tend to be associated with higher risk of certain diet and lifestyle related cancers, whereas consumption of more unprocessed food is potentially protective against these cancers. To quickly move on to some of the more kind of biomarker molecular aspects of the work we've been doing. So I'm sure many of you remember some of these kind of metabolic pathways, which are many of you studied at university or medical school. But what we're particularly interested in is trying to probe more deeply metabolism and identify metabolic markers and how diet interacts with endogenous metabolism, which then can be linked to different cancer outcomes. So different tools such as metabolomics are now being applied within large prospective studies, which can give a very broad but also detailed snapshot of endogenous metabolites, but also exogenous compounds within blood samples collected within cohorts. And these can be related to, for example, different aspects of the diet and body mass index, activity, also the microbiota, and can also then be linked to cancer risks. So within EPIC, we've been applying metabolomics not only to help refine or identify biomarkers of dietary exposures, as well as other aspects of diet and lifestyle, but we've also been linking some of these metabolites and metabotypes to cancer risk. Just to give an example of how we've been applying metabolomics within the field of diet and cancer, I'm going to give a very quick overview of some work we've been doing on red and processed meat. So the role of red and processed meat in cancer is very controversial. Several years ago now, the IARC monographs classified processed meat as a group 1 carcinogen, meaning it is a cause of colorectal cancer. And red meat is a probable cause of colorectal cancer. And this was based on epidemiologic evidence, as well as some degree of mechanistic evidence from both animal models, but also in vitro studies. Obviously, the consumption of meat is and its relationship with health is an area where there's a lot of interest, but also controversy. And several years ago now, there was a new set of meta-analyses which claimed that there was actually no association between red meat and cancer, based on the fact that there's no clinical trial data on this particular exposure, which we know is very difficult to do. It's very difficult to do randomized controlled trials for dietary exposures such as red meat, and also with a cancer outcome. So these sorts of evaluations have always relied on high quality prospective data from cohorts with triangulation with mechanistic data. And I think one of the limitations in this field in particular is lack of good biomarkers for red and processed meat, as well as lack of mechanistic evidence. So the work we were doing at IARC to try to identify new biomarkers of red and processed meat, this was work that was led by Roland Wedekind, who was a PhD student at the time, was to do a dietary intervention study of red and processed meat, and try to identify novel biomarkers of red and processed meat, which could then be related to cancer outcomes in EPIC and other cohorts. So I don't have time to go into the details, but essentially this was a study where they had the randomization of 12 healthy outcomes to healthy adults to different diets with different types of processed meat, so hot dogs, bacon, and salami, a non-processed meat, pork, and then tofu, which was a control. And then they collected blood samples from participants and urine, and then ran high resolution mass spectrometry to identify compounds within the blood, which changed when consuming these different types of meat products. And what they found was that there were certain compounds found within blood and urine which were correlated with intakes of these different types of meat products. So this 4-methylsyringol sulfate, which increased substantially consumption of hot dogs, but also bacon to some extent, but less so for salami and pork, and piperine, which is another compound which was found to increase upon consumption of salami. The results were then validated in EPIC using cross-sectional data, so taking the blood samples from EPIC and then looking to see whether these biomarkers were associated with reported intake of different types of meat, and they quite convincingly were. So, for example, this 4-methylsyringol sulfate, high consumers were also found to have high levels of relatively high levels of correlation with different types of processed meat within EPIC. What we're doing now is trying to relate these biomarkers of processed meat to cancer outcomes. This is work we're doing on colorectal cancer. These are very preliminary results, but what they suggest is that there are certain biomarkers of red and processed meat which are associated with colorectal cancer, but it seems to differ depending on subsite of within the colorectum. So, for example, this piperine compound only seems to be associated with proximal colon cancers, whereas hydroxyproline was possibly associated with distal colon cancer. So, suggesting that maybe some of these biomarkers could also help disentangle the effects of where processed meat is acting within the colorectum, and if validated, could be interesting biomarkers for processed meat in going forward. To quickly show some work that Pietro led looking at alcohol biomarkers. So again, alcohol is a very important aspect of diet and lifestyle. It's been linked to different cancer outcomes, but until recently, we've not really had a good biomarker of alcohol intake, and we know that people often misreport alcohol consumption. So work that Pietro led with colleagues at NCI identified a novel biomarker of alcohol consumption, 2-hydroxy-3-methyl-butyric acid, which was shown to have a much better predictor of liver cancer and pancreatic cancer compared to just relying on self-reported information on alcohol intake within the cohort, both in EPIC and also in the ATBC cohort in Finland. So again, just demonstrating some of the power of using these kind of metabolomics approaches to identify biomarkers and then relate them to cancer risk. So almost out of time, but I quickly just want to touch upon obesity and cancer, because I think this is a really important topic around diet and lifestyle. So again, over the last couple of decades, there's been a wealth of epidemiologic studies which have looked at the link between being overweight and obese in relation to cancer. The IARC handbooks evaluated body fatness back in 2016 and judged that there's convincing evidence that obesity is a cause of at least 13 different cancers, so which are listed here. You can see quite quite a variation in the degree of magnitude of association between obesity and different cancer outcomes. So very strong associations for endometrial cancer, which I mentioned earlier, esophageal adenocarcinoma, but much weaker associations. So, for example, for postmenopausal breast cancer and even colorectal cancer. But given that these cancers are very common, the population attributable fraction of obesity to the burden of these cancers is actually could be quite considerable. And given the ongoing obesity epidemic worldwide, and the growing rates of overweight and obesity in many different regions of the world, given this link between obesity and cancers, this could be a growing source of cancers in the years to come. But the important question is, what can you do about that? So obviously an obvious answer would be to implement more public health measures to try to avoid obesity becoming prevalent in society. And I think a number of different countries are trying to tackle this. For example, sugary drinks tax, trying to encourage more physical activity. But that might not be enough, given the scale and upward trend of the obesity epidemic and the growing number of cancers linked to obesity and the growing cancer burden. We might need to have more directed interventions to try to break the link between obesity and cancer. And really, to do this, we need better understanding of the biological mechanisms linking obesity with cancer. So again, this is an area where there's been a lot of research activity in recent years in trying to understand why obesity is a cause of cancer. What we're currently thinking is that obesity engenders this kind of physiological, endocrinological phenotype, which means that cells which bear oncogenic mutations are more likely to survive. For example, because of the actions of obesity increasing insulin, insulin-like growth factors, which increase proliferation and suppress apoptosis, but also the role of inflammation and sex hormones is also quite likely an important factor. But there's still a lot we really don't know about the link between obesity and cancer, and I think this is an area where there's a real, growing interest in trying to identify what are these links and what can we do about this? Can we actually use this information to prevent cancer among obese individuals who are more susceptible to developing cancer? Just to conclude, so key messages I would say are really, so I think large-scale, carefully conducted prospective studies of nutrition and cancer have yielded results that support laboratory data, and to some extent in some intervention studies, which I've not been able to get into today, but there have been some intervention studies around diet. For example, Women's Health Initiative, which showed that, for example, women who are consuming more dietary fat had a borderline increased risk of breast cancer. So that's somewhat consistent with what we're seeing in prospective studies. But what we need going forward is more high-quality pooled meta-analyses, so trying to increase sample sizes. As I said at the beginning, we're looking at very weak to moderate effects. So having large sample sizes and adequate power to link diet with cancer, I think is going to be crucial. And then future work will be needed around identifying more precise aspects of the diet, biomarkers, temporality. So most studies only really look at diet in midlife. And it's quite likely that dietary exposures and endogenous exposures early in life are likely to be playing a role in future cancer risk, but studying that in current context is actually quite challenging. Integrated nutrition, so moving away from single nutrient hypotheses, so looking at dietary patterns, bringing in some of the newer aspects of diet classification like ultra-processed foods, food biodiversity, I think is a really interesting and novel area. And also then bringing in genetics, so looking at gene-environment interactions. So some of the large genetics consortia, for example, for colorectal cancer, are now seeing that the association between diet and colorectal cancer is modified by certain genetic variants, which can itself give clues to mechanisms. And also using tools like Mendelian randomization can also be really valuable in this context. And then very lastly, most data currently that's being published comes from the US, Europe, Australia, and Asia. And we have very little data from Africa, South America, and other parts of the world. So, IARC is really committed to trying to develop more cancer research within these regions. And I think this is going to be really important for diet and lifestyle, because we know that many countries around the world are undergoing this epidemiologic transition, where they're moving more towards Western lifestyles. So it's quite likely that the burden of cancers attributable to diet and cancer is going to increase in more low to middle income countries in the future. So I think more research in this domain is really going to be important. And I'll stop there because I know I'm over time. So sorry about that.
Thank you, Mark. That was really interesting, as usual. Always learning something from your talks. So now we have a question and answer section. We have a few questions from YouTube and some questions in the chat box here. I would ask Neda, please, to ask your question. If you can open your mic and camera, please.
Hello, everyone. Here, Professor Marc Gunter and Pietro Ferrari. Thank you so much. I have some questions about the relationship of diet and nutritional risk factors of cancer. In my literature review regarding our project, there was a inverse relationship between obesity, body fat, and risk of breast cancer in premenopausal women in Latin America. In my opinion, I think that because of the low food security, they give the adequate nutrients in the charge of eating more. For example, they is this my opinion is true, or what other explanation of this association?
Yeah, so thank you for your question. So you're raising a very important point that while being overweight and obese increases risk of breast cancer after the menopause, for premenopausal breast cancer, being overweight reduces risk. We don't really know why that is. Is a blunt answer. I mean, most studies around the world show the same thing, that being overweight earlier in life reduces risk of premenopausal breast cancer. There have been some suggestion that kind of hormonal changes around the age of menarche and body size in kind of the earlier adulthood has certain hormonal effects which could potentially prevent breast cancers. But we really don't know the answer to that. It's a really interesting phenomenon. And I think one we don't really have the answers to. So there is a lot of research now ongoing to try to identify causes of premenopausal breast cancer. But yeah, I don't think we know. So we really need more research in that area.
Okay, thank you, Neda, for asking your question. Mark, we have another question from Kamal. Kamal has poor connection, so I'm going to read his question. He's asking, does physical activity helps to improve the quality of life of cancer survivors?
Again, good question, and I would say we don't really know. So I didn't really touch upon cancer survivorship because the focus is really on kind of cancer development. But yeah, I mean, there's a whole field devoted to the impact of diet, lifestyle, and physical activity in cancer patients and how that relates to survival and prognosis. There have been some suggestions that people who are more physically active after a cancer diagnosis have better outcomes, particularly for breast and colorectal cancer patients. But I don't think that the evidence is really strong enough to be judged as convincing yet. This is an area where, particularly for colorectal cancer, there's a real lack of robust evidence in that area. But there are studies suggesting that being more physically active at the time of cancer diagnosis and afterwards is associated with better outcomes among those patients. But I would say that the evidence is not definitive currently.
Thank you, Mark, and thank you, Kamal, for a good question. Iriwan, please, if you can open up your mic and camera, and yeah, we have a few questions to go, so please be short if you can. Thank you.
Okay, I have 3 questions if we have time. Thanks for everything. My first question is, so I have only one.
Go with the first. We see that.
Okay. Risk
Thank
of
you.
colorectal cancer and vitamin D deficiency, do you think that, as far as you know, could nutrition deficiency lead to also cancer, apart from excessive consumptions?
So vitamin D and colorectal cancer, you're asking?
Yes.
So this is another area where there's been a lot of controversy. So a lot of prospective studies have shown that people consuming higher levels of vitamin D through the diet have a lower risk of colorectal cancer. And there is some mechanistic evidence to support that. But I think just recently, some of the evidence has been thrown into doubt from coming from some of the Mendelian randomization studies. So using genetic proxies for vitamin D exposure, which were did not support a causal association between vitamin D consumption and colorectal cancer. And we know that relying on questionnaire-based data for vitamin D, and even to some extent biomarker data, there's always chance of confounding because we know there's vitamin D is correlated with other aspects of the diet and lifestyle, which can potentially link to colorectal cancer. So, and certainly the randomized controlled trials of vitamin D supplementation did not show a convincing effect on reduction of colorectal cancer. So I would say that currently this is another area where we don't fully understand the link between vitamin D and colorectal cancer. It's somewhat suggestive, but it's not definitive.
Thank you, Mark. Iriwan, please, you can ask another question if you want to.
Okay, thanks for your answer. You concentrated on the amount of the meat consumption, and you partly just said types of smoking or bacons. I have a question: is the type of the animals based on either industrial farms or based on organic? Is it being included in the studies, or just the amount of the meat consumption is included, and the variations in the culture regarding cooking, types of cooking of the meat, like barbecue or cooking, does have impact on the association and linkage with cancer? Thanks.
Yeah, that's a very good question. So currently, nearly all the studies just look at total amount of meat being eaten. They don't differentiate between whether it's factory farmed or organic. Remember, a lot of these cohorts collected this kind of information 20, 30, 40 years ago, where there was less organic farming. So I think some of the newer studies are looking at that. They're trying to incorporate more data on whether the food was organic or whether it came from kind of more factory farming practices. But currently, we don't have good data on that distinction now. But I think you're right. The quality of the meat and how it's cooked and prepared is probably going to be really important. We know some of the compounds which have been which are formed in, for example, red meat when it's cooked, have been linked to cancer, and probably the method of cooking is probably relevant. But again, we don't really have good definitive evidence on that yet. So yeah, I think that your question is a really important one. I think it's an area of current research interest, particularly around organic farming. There's a lot of interest now in the presence of other compounds within foods, which might come from pesticides or factory farming or other aspects of how food is produced. I mean, I touched a bit upon that with some of the ultra-processed foods. So we know that when these foods are produced, they have a lot of compounds added to them to preserve freshness and shelf life. And we don't really know how those compounds might be related to cancer and other health outcomes. So I think in the coming years, we'll see more high-quality research in this area, which should be able to shed some light on the questions that you asked.
Okay, thank you, Mark. I'm going now to read a couple of questions. So there was this question about, well, along the same line, is whether plant-based diet is more protective against cancer risk than animal-based diet. is it a myth or truth what research suggests?
Well, we don't know, I would say. There is a lot of interest in now in these kind of plant-based versus animal-based diets. We're actually doing some work on that in EPIC at the moment. There are some studies suggesting that consuming a more of a plant-based diet may be associated with lower risk of certain cancers and disease outcomes, but I would say that's not definitive. There's good reason to think that should be the case. We know that consuming more vegetables and fiber are likely to be beneficial for reducing risk of certain cancers and also other chronic diseases. But yeah, we don't have good evidence that following more of a plant-based diet reduces risk of cancer. Yeah, again, this will be an area where I think there'll be more high quality research coming in the years to come. So I would say at the moment, it's not a myth, but it's not proven that is the case.
Thanks. Neda, you're very keen to ask another question, please.
Thank you so much. In the case control study about the dietary risk factor of the cancer, for example, we design a case control study as the nutrition and diet. The cancer itself disturbs the nutrition and diet. So when we perform a case control study and to compare the dietary intake of two groups, there is a question that this dietary intake is before the cancer or cancer itself disturbed the nutrition intake. For example, the cancer disturbed the activity daily living and the capacity to go buy and prepare food and so on. What can we do in these situations? And another question, would you please tell us about the Latin America cohort studies that are going and have enough data about performing nested case control study? Would you please suggest the best one to select and design a nested case control study upon that? Thank you so much.
So your questions about the problems of looking at nutrition and diet in case control studies.
Yes.
So that is a perennial problem of those sorts of studies. So we know that when you recruit cases into case control studies, they already have cancer. So they might have already modified their diet. So not only at the time of their diagnosis, but also maybe in the years before they were diagnosed, they were starting to get sick. So they've changed what they're eating, which might not reflect their habitual diet from many years beforehand. So we know that there is that bias. And I think it is a problem for case control studies of diet and cancer. And that was really what led to many of these big prospective studies being set up, because researchers are very much aware that case control studies were probably wholly inadequate for studying the link between diet and other exposures as well, but also particularly diet and cancer. So there isn't a good way around it. Even relying on blood measurements is problematic, because we know that once you've got cancer, it affects your metabolism, and there's also a bias in relying on kind of biological measurements within case control studies. So for many countries, some of the ones you've alluded to, we currently only have case control studies, and I think they're important to do for several reasons. Firstly, they can give us some clues as to the link between a particular aspect of the diet and cancer, even though we know there's biases and there's limitations. As long as we judge the results of those with those limitations and biases in mind, they still can be useful. But ideally, we'd want to set up prospective studies in low to middle income countries, including Latin America, which you've referred to. So to my knowledge, there are no prospective cohorts in Latin America. There's many case control studies that have been conducted, particularly around diet and cancer. I think those case control studies are valuable. As I said, they can offer some clues. They can tell you a bit about the prevalence of consumption of certain aspects of the diet in those populations, but they're very difficult for kind of ascertaining potential causal links. I mean, prospective cohorts as well, I've got their problems, and also cannot ascertain causality, but they can do. We can remove some of the biases inherent of case control studies by performing prospective cohort studies. But anyway, I hope that answered your question. I think the bottom line is diet is tricky for case control studies, and we currently don't have any prospective studies in Latin America, so we have to rely on these case control studies until there is one set up.
Okay. Thank you, Mark. It's 2 p.m. I'm allowing just one last question from YouTube for you to answer, please. So the question is, how stress derived from obesity could have an impact on obesity-related cancer?
Stress. Yeah, it's interesting. I mean, the role of stress in cancer is something that there is growing interest. There's some experimental studies suggesting that some physiological impact of stress could play a role in cancer development. It's quite difficult to study in human studies, and particularly in cohorts. I mean, we didn't necessarily collect information on stress in EPIC and other cohorts. And also, stress is a very subjective exposure. So what might be a stressful situation to one person could be different to someone else. So asking people about stress, I think it's worthwhile doing. What we probably would need would be kind of biomarkers of stress and how physiologically we respond to stressful situations. Obesity, yeah, I mean, I think we don't even really fully understand the link between stress and cancer, let alone whether it might be an intermediate or play a role in the obesity-cancer relationship. But it's certainly possible. But yeah, I think it's a good question.
Great, and a very great answer that made us understand once more how difficult it is to disentangle the role, the impact of different players in even in one single relationship, because so many different factors could intervene.
Yeah.
Now, please join me in thanking Professor Marc Gunter again. Netu, Inas, Betul, apologies. You asked very good questions, but no time, and I wanted to give room to everyone.
I saw Pietro just the last question from Betul. I think we can answer that quite clearly. No, okay. There's no definitive anti-cancer food. Yeah, if you ever see things in shops saying this is an anti-cancer compound, there's certainly studies which have shown that certain compounds, such as the ones you've mentioned, like turmeric, they potentially have anti-cancer properties in experimental models, but the human data is not reliable, and it's very inconsistent. So until that's been shown in a randomized clinical trial or an equivalent, I'd be very wary if you see something being called anti-cancer. So maybe that's a good note to end on.
Okay, good. Yeah.
Just to say, people are welcome to contact me by email if they've got, I didn't get to answer their question, or yeah, I'm happy to follow up.
Great. And then, yeah, I can tell you, if you send an email to Mark, you will get an answer as a fact. So
I try.
Thank you, Mark, again. That was super interesting as usual. Okay, and
thanks everyone.
Thank you to the audience. Yeah.
Good luck with the
course. Thank you very much.
Bye.
Thank you so much.
Thank you so much.
Thank you very much.
Yeah, thank you so Mark, much. See you soon. And for our viewers on YouTube, the next public event will be on the first day of the face-to-face in the epidemiology module on the 19th of June, same time. And there again, we'll talk about social inequality.